and the two Frenchmen who were pioneer in discovering something called the lactose metabolism, glucose galactose metabolism inside the model organism called E. coli, had this famous statement, okay, because they were very astute and ahead of their time. They did not just focus on genes and enzymes and chemistry and compartmentalized, but this statement was really, foreseeing that it seems likely that the divergence results not only just from changing, but regulatory circuits rather than chemical structures. Okay, so they were, forecasting the advances of this micro or nano biology that we were. And this quote, I wasn't aware of it, but our friend John Hassel pointed this out to me. And that's his book, that he has incorporated. So I have to thank him for this. And again, I may have mentioned in the past, nature is very conservative. It is, the same chaps, Jacob and Mona, okay, who also said what is true for E. coli, the smallest work organism, is true for elephant. And so all these processes are conserved, from on evolutionary base, some of this circuit. So we'll talk a little bit more about it, as to this. And a lot of this discovery was done by a rather eccentric Englishman who went outside the laboratories and universities. Fortunately, he was wealthy enough to support his own research. He was assisted by his able lab assistant, whose name I forget, and he was instrumental in incorporating this mechanism of proton and electron transfer and chemiosmotic theory. That is the name of this incorporation of physics and energy and biology. So, and on the other hand, one can say he upset the whole apple cart of chemistry. And this, again, was one of those famous controversies in science. He had proposed this idea based on his pure power of thinking alone and was very revolutionary. And like it happens with many revolutionary ideas, it was not easily accepted. And it took about 17 years from 1961 to 78 before this could be proven that he was spot on and he was correct. And later on, he was richly awarded by a solo Nobel Prize, which is very rare in modern science. Okay, so that's, the eccentric but a genius, so Peter Mitchell, who is less well known than other like Watson and Crick, but perhaps he should be because he incorporated various disciplines. I've already talked about the charge of DNA and other stuff. So now, this was, some of you who are science aficionados may remember this, 2001, simultaneously when the Human Genome Project was completed, both magazines in Britain, Nature and Science in the United States, came out with the completion of Human Genome Project. Now, genetics is well known. A terminology was coined way back in, the early 80s, and then it became genome, means the science of genetics. And then obviously we know that proteins are the workhorse of biology, so it became proteome. And then we came up to idea that we have lots of microbial flora inside our gut, starting from the acid ulcer-causing bacteria H. pylori to other huge number of microbial that help digest our food, so microbiome. So the same way, now a new term has been coined and come into vogue. It's called electrome, okay, which stands for the conversion of, okay, yeah, junk call. For currents in living entity from cell to organization, okay? For example, when I raise my hand, obviously the impulse travels from my brain to my muscle. And so that's, it's an organizational level. So one can say it's an electron. So it's a mechanism of sender and receiver, and this again transfers information, which, information itself is, does not have any mass, but it's just the ways of passing on the signal. So this is all is needed for organizational level. And just today, by sheer coincidence as to how information is valuable, on the BBC website, there is a lucky or unlucky woman who had a train accident, who lost her arm and a leg in a train accident, on the London tube. And unfortunately, she was able to receive AI-inspired artificial prosthesis. And usually with the prosthesis, people have to actually think and be able to move. That was the state of the art. But with AI, the system, the prosthetic learns by itself. And now she can literally cut an apple without having to think about it, type of thing. So it's a phenomenal thing about how, the integration of computer science, uh, bioelectronics, and biology has incorporated. I mean, it's just, I shared it on my social page for Facebook type thing, but you can just go to the BBC website and see it. But as to how all this interdisciplinary conversion, okay. And again, like we talked about last time, when you die, what happens? The body temperature drops. That's how the coroner determines the time of death on any crime shows. And so death is when loses heat, because the system is not conserved, heat is dissipated. In other words, to use a physics term, entropy, increases. So that's, when the body starts cooling off. Okay, so enough philosophizing, but okay. So this is, I'm sorry, let me see if I can move back. Okay, yeah, so biophysical essential of immaterial dimension of life. So that's like some philosophical axiom. Okay, now, yeah, and I'm obviously, all of you are familiar with in the beginning was the word, but when we get down to it, in the beginning was the potassium channel, because when obviously, life evolved in ocean, and the flow of current that was going through a single cell, which was this salt inside salt water. It was potassium that was inside the cell, so it was an electrical current was set up. The gradient difference with the ions was, so one can say in the beginning was the potassium channel. And I just found this yesterday, as to how the porpoises and cetacean life, all these channels are conserved. These are there inside whales and porpoises, and life. So in all the system, all these channels are conserved that you and I have. In the same way, the importance of potassium channel, you can see it's at all levels, starting from and these are the names are not important, but you can see starting from your brain all the way to your testicles and bones, have a very diverse distribution. So, okay, so in other words, we owe our activity of each and every cell to influx and of sodium and potassium interchange that takes place. Okay, and similarly, you can just see, let me see if I can zoom in on this one. It's at the various levels, all this fancy, there are about hundreds of channels that have been discovered, and the names are only for the people who are really into science, but starting from vision onwards to your brain, to, walking, stride length, and last but not the least, the most energetic organs that we talked about, the brain as well as the heart. Okay. And I just want to make sure that we are recording this, right? Okay. Oh yeah, we are. Thank you. I almost had a. Okay, it's worked out nicely so far, so I don't have to re-record it. Okay, and these are the varieties of ion channels, just for people who are more curious about science. And these are the types of ion channels. It depends upon voltage. Ligand is the small protein light-gated, okay, mechanosensitive in simple touch, because in the beginning, the complexity was just simply touching the another cell, okay. Nucleotides are the A, C, G, and T, okay, inside your DNA, and calcium, last note, calcium, even though it's not as widespread in ocean water, it is still equally important, as we'll see when we talk about. And again, this may be interesting that even the in sex, the simple touch in the genital area, the pressure, since it opens up this piezoelectrical mechanosensitive channel, okay? And as you can see that the pressure, it turns into ions and forms a current, okay? So it's so it's interconnected, how it's nicely ties up, okay? You can just see how, and this is the principle of ultrasound, by the way, which is what I've done most of my professional career, was ultrasound and electrocardiography. So this is how we have imitated the idea from nature as to how there are certain elements, which can, respond to pressure and generate a silica type tissue that we have used as uh, doing ultrasound. Okay. Okay. And so there are, tons of channels, and there are various receptors I've talked to you about. There's, that the nicotine receptors inside the body, and there are various other receptors, which we'll talk about, that's something very interesting for movie aficionados will love it. Now, why is the ion channels, why did they evolve? Because, there is nothing to take away from DNA and everything, but DNA replication, that takes its own time, because it's they have to come up with the right genes, and from 3 billion letters. So varieties of genes it's a complex orchestra of hundreds of players, so to speak. So it's a time-consuming process. So the gene, you can see from the time, DNA replication, cell division, it's time-consuming. Gene transcription and protein synthesis takes about an hour, okay? And hormone regulation also, it takes time. Enzymes are quicker, but the ion channels and vision and everything else, I mean, you can just see that's why baseball player can react so quickly, because their brain has already sensed before the ball is released by the pitcher, and they've positioned themselves to hit a home run. So this is milliseconds. So the action is amazingly fast, as to that gives us the quickness of response, and before the adrenaline pumps out from the adrenal gland, the fight and flight. So electricity is what makes the world go around. I mean, nothing to take away from adrenaline, the stress hormone, or dopamine, the pleasure hormone. Okay, but electricity, that's the reflex that we talked about when we talked about muscle. Well, tapping your ankle is just enough to do it. Okay, all right. Now, obviously, all of you are of Christian faith, or at least were, and you may recognize this, quote, okay, which, now, obviously, you can say it sometimes, for the so-called, some of these miracles, we can sometimes we get lucky and find an explanation. This is, obviously, all Floridians recognize the red tide, okay? But there's a detailed and the warning sign, as you can see, a common occurrence in Florida, red tide sign, okay? And now this is the great chain of, not chain of being or life, but the chain of toxin. There are tiny microorganisms called dinoflagellates, that are consumed by the shellfish and the corals, and it gets inside the fish, and small carnivores, large carnivores, including humans, so that toxins can be transferred. And that is the one that creates the red tide, okay? And that's the biotoxin that was evolved because of sunlight reactivity, it was evolved from the tiniest organism, okay, and it transfers via the food chain. Okay, let's see. And again, that works via, this has been chemically isolated. It's a chemical name is, for those of you who are curious, it's called saxitoxin. Okay, so nature has used these chemicals for uh, defense mechanism. And as you can see from this slide here, that the channels and potassium is enters, toxin blocks the channels, and hence the death of the uh, enemy organism. So you can see how the toxin is just has the right shape it manages. So this is, and so that may have been an explanation for the red tide and the biblical, you know, uh, ness. And this is again, it's a recurrent problem in Alaskan mussels and the uh, fish population. And there is, it's a very small amount, but when it accumulates, when you eat substantial enough portion, it can be very fatal. Now along with the toxins by one, other organisms form a defense mechanisms. Some of the clams, have developed another mutation inside them, so that they do not die, but it saves the heart even when their respiration is paralyzed, so to speak. And again, that as human beings, we know how to use and abuse science as it was done, during the First World War with nerve gas, which again, interferes with these uh, channels and respiratory channels. Same way the U.S. military has stockpiled saxitoxin toxin for warfare. Okay, and I don't know if they've destroyed the biological warfare this thing or not, but that will still remain a secret. Okay, so this is how things are connected with. Okay, and I alluded to the other common toxins, which is the Botox, which is it can be the botulism poisoning can be fatal, but extremely micro doses. It's a big business nowadays, for paralyzing your wrinkles and reversing aging process, but it wears out in short time, and there can be some side effects. And the same way, the native of South America had used this for a long time for hunting and for tribal warfare. And the curare poison blocks the workhorse of neurotransmitter called acetylcholine. And one one of the worst Nobel prizes was for nerve gas during First World War when it was released. Okay, and so this is again now, and it's not all this thing. The scientists have used this as a model in micro doses in researching these ion channels as well. So poisons can have its use, and obviously people have been using it to create antitoxin for snakes and other organisms. I have some slides coming around. And some of these toxins were used as poisoning during Second World War to kill, as an attempt to kill one of the Nazi officials named Heydrich in Eastern Europe. And the US military has attempted to use this for Fidel Castro. Obviously, they did not succeed. And inhibition of the enzymes that breaks down acetylcholine, that's the way the poison acts. It leads to convulsion and deaths shortly thereafter. And this has been around in various other cultures as well. I went to med school in India, and it was very common to use a plant called datura, Datura. The seeds were used for suicide attempts. Okay, even and this is a historical link that even in Gandhi's biography, when he was a kid, he had done something that he thought would upset his mother. So at that time, he went and took some datura seeds, but didn't take enough dose, so didn't die, but finally confessed and went on with his life. Otherwise, the history of the world might have been different, perhaps. And the same way, the same type of natural beans called kalar bean are used in Nigeria as well. So human beings have used plants for poison and for hunting purposes for a long time. Okay, so by trial and error, they had figured it out. Okay, and these are some of the other lesser known poison called aconite poisoning. This is the plant to which called wolfbane, the botanist or the uh, gardeners may recognize this. And there are other poisons which are, the names are academic, but this little tiny dart frog packs a whopping punch that, about, and the nature just really, why do they need to develop such a lethal dose? That's just beyond me. But and the same way, there is, honeybees develop some, uh, gyanotoxin. Okay, and there is something called the mad honey disease, which comes from toxic honey, from rhododendrons. Okay, it can be fatal. And rhododendrons were discovered, I think, in the Himalayan foothills, and they were imported by the Brits and became worldwide. And also the DDT had the similar effect. Okay, so this is, the poisons have been around from 400 BC. The Greek author Xenophon described this in his expedition to Black Sea. Okay, and we already referred to Botox and other curare poisoning. So these are all the so-called sodium channel poisons. So and I think I mentioned briefly about in the Agatha Christie book that and the king of it all was the Japanese, the puffer fish, or as the Japanese name is called, fugu, F-U-G-U-E. It's a weird looking fish and has been known since the Egyptian era, but still very popular in Japan. It's considered a delicacy, and it consists a very powerful toxin in its liver and ovaries. Okay, and the Japanese, they're very organized society, and it's highly regulated that you have to have a special training to be a fugu chef. Okay, and again, there was a famous Japanese actor who insisted on eating the liver, and because he was a celebrity, he was served and he died after that. So again, it's even more tightly regulated, and still about half a dozen or so deaths do occur even in Japan with. Okay, and this can be useful to develop drugs for pain relief, because it stops the action potential. So one guy's poison, one species poison is another medicine. Okay, and it is possible that Captain Cook, suffered this as well when one of his trips, they had eaten entrails of the pig that had this type of toxin. Okay, and similarly, this is how your local anesthetics work as well, by the way. Okay, so this is the famous tetrodotoxin. Okay, and not only that, other animals have evolved this poison as well. Okay, you can see this is, on the upper left is the puffer fish, and this is a tiny little slug, and it has evolved the same poison. Okay, and this cute looking octopus is a tiny little octopus, blue ringed octopus, but it's severely toxic. Octopus in general are not, but this little species is. This is a three-colored frog in South America. Same way, this is a rough-skinned newt. And last but not the least, the horseshoe crab. Okay, and so all of this, so it's widely used for, has evolved and many times in different species and genera. Okay, so what to be Now, I don't know if I mentioned this or not, but it is really interesting to know about the fainting goats that were imported in Tennessee sometime in 19th century. And this has, again, there is a genetic mutation of simple one amino acid that has taken place, and that causes just a tiny little shift in the chloride channel of this poor little creatures. Okay, and that creates less open channels, which is near the skeletal muscles. Okay, and so when they get these goats, when they get excited or frightened or whatever, they just faint. Okay, their muscles just tighten up. Okay, so it gives you the idea of the molecular or genetic basis of this illness. And these goats have been selectively bred because they can't run away. They just faint when they start running or excited, so that they're easier to hurt. And they have bigger muscles as well. So for those of who are using it for food and meat production, they've been cultivated. Okay, and they can't jump over a fence basically. Okay, so that is a big business for in a certain part of farming community. Okay, so the faint, and there's a later on, if I have time, I have a video for this, but you can just see how that. Okay, and again, this is not just, the list is far and vast. Diseases that involve ion channel function, starting from a simple dry mouth to anxiety to, we'll talk a little bit more about cystic fibrosis. And again, you can just see diabetes, multiple sclerosis, ringing in your ears, various. So this has been involved in the classical one being epilepsy, which we'll talk a little bit more about it in a few slides. Okay, and again, and these are not just that, we have used varieties of drugs as well. So you can just see that some of the drugs that you recognize, diazepam, which is good old Valium that came around, the glyburide, that diabetic medications, basically, and these are various heart medications. And I mentioned the calcium channel blockers, which is your nifedipine and procardia that some of you might be taking, or cardizem or diltiazem that you might be taking, and all the anesthetic that we talked about, phenytoin, which I'll talk more about it. It's an interesting human interest story. So varieties of pharmacopoeia depends upon ion channel modulations. Okay, so so there, and so the role of this thing starts from anywhere, right from conception to taking a deep breath to forming, organs to immunity. So it has a widespread use, basically. Okay, and okay, and not only that, it is also useful in the botanical this thing, because southern corn leaf blight, which occurred in 1970s, okay, that had to do with abnormalities and poisoning by fungi on the plant growth, okay, and messed with the potassium channel of the corn plant and caused a huge amount of economic and crop loss. Okay, and so this is, again, and this is used by various animals as well for protection, such as the Venus flytrap. Okay, so you can just see that it has a widespread role from plants to tiniest bacteria to various organisms, and not last but not the least, human beings. Okay, and now this is, again, the Nobel winners go through various fashions. I mentioned that in the early 20th century, the neurology was popular. The second half of 20th century, DNA and various genomics thing became popular. And at the same time, ion channels were getting more attention as well. Okay, and this was a scientist who received Nobel Prize for discovering protein. Only water may pass. So there is a special chain. So water being the most important molecule of life, and you can just see that how this is called the aquaporin channel. Okay, so how based on the uh, water molecule that rotates and it can pass. It's called aquaporin channel. So this is, this is has been a very important in use of biochemical and genetical studies of various proteins at a very molecular or cellular level. Okay, and this is again has a huge role in various diseases of kidneys and muscles. Okay, and this has been a floodgate has opened up, so to speak. Okay. Okay, and various organisms that hibernate have a role in they can shrink their blood volume in winter without causing cell death. The tiny little shrew that freezes. So certain animals have evolved dramatic of defense mechanism to survive harsh elements. Okay. Okay. Now again, this is as you may recall, I did a class on senses, and I had sent out the slides of the use of electricity in sensing in whales and other things, but also the biggest part of vision. We are all human beings are visual creatures. A huge area, one third of our brain, is occupied for processing vision as to what and where, how to process the information that we see. Okay, and the ion channels have a huge role to play because this is a direct mechanism of converting light into electricity because and the photons that we received through our lens, were through various layers and a molecule called rhodopsin, which is, by the way, a derivative of vitamin A. I'm sorry, just looking. Yeah, we have time. So we're moving well right along. It has a lot to do with the formation of this protein. Okay. And it's a the eye is evolved backward because the mechanism that the retina is in the back. So it's why that evolved in such a way because the nerve fibers are way at the bottom, not in the front. Okay, that's different than octopus, and so nerve fiber layers are way behind. The retina is in the front, the pigment. Okay, and again, you can just see all this uh, alphabet soup includes varieties of channels that include the potassium and the touch channel, the TR and calcium channels. So these are all in the retinal epithelium. And there are various photoreceptors. That's the protein that changes its shape. It connects to the other interneurons. And then finally, the impulse is converted into electrical signal, okay, and gets down through the nerve fiber, okay. So this is, the light is transferred into electricity, okay, and even, it's extremely sensitive that even the tiniest amount of photons will activate this. Okay, and the center of the eye is called fovea, as the cones and rods that that you learned in school. Okay, okay, and again, the rods and cones have to do with color vision, and again, color vision evolved later on in different species of animals, and you know and it's but again, I don't want to digress too much into evolution, but this has a lot to do with it, and that has been a big gift in improving the nutrition of getting more glucose with being able to see the right fruit and getting more energy in various chimps, and the evolution of that was different between the old world and the new world monkeys. So the color vision may have evolved more than once, okay, in different uh, chimps and different monkeys. Okay, so this is, the basics of the visual system. Okay, and again, this is, just illustrates the main point, the opsin protein, which comes related to vitamin E, and there are varieties of receptors called. Okay, let me see if I can. Okay, gets around and then it changes its shape, and finally, it converts into electrical signal as it goes around. Okay, so it's a very complex mechanism. It involves varieties of channels, light, and finally transmission, into receptors and to the nerve, through the nerve fibers. So, okay, and again, the basic enzymes, the instead of ATP, it's a glutamic monophosphate. It's converted, and the same type of influx and efflux of sodium and potassium takes place. Okay, and again, nature reuses vitamin A, recycles, just like it does, in the glucose metabolism inside individual cell. In eyes, it recycles the vitamin A. And some of this research was, the vision mechanism was completed by two brave scientists who were torturing their cats, but they discovered how vision evolved. The centers around as to how, the varieties of cells are oriented during early evolution. They taped the eyes of a cat and discovered and recorded the nerve signal as to positional orientation. So it's a very fascinating piece of research that was done, by using tiny tungsten electrodes. Okay, excuse me. So let me, on the screen as to how the optic nerve was, the stimulus, the directional interpretation in various parts of the brain, it took place as to how, so this was their 1959 groundbreaking discovery, and rightly so, they were awarded Nobel Prize in 1981, if I recall correctly. Okay. Okay, those of you who are country music or pop music fan may remember this song. Why did I put that in? Okay, the charts are not blue like this. This has to do with the popular medicine Viagra. Okay, that sometimes, it inhibits the enzymes in your eye, called phosphodiesterase. Okay, and that can alter the function of retina's photoreceptor cells inside the rods, okay, which are most sensitive to blue light. So this increases the sensitivity to blue light, causing the objects to appear tinted blue. Okay, I mean, it's not really a major this thing. It's usually it's a self-limiting. Okay, thank you, Donna. Okay. Okay, so it has, but okay, let me see. Yeah, so it's a very relatively weak inhibition. So you're not going to lose your eyesight, if you overdo it. Okay, as your grandma might have warned you, or your clergyman may have warned you. But it works with the cyclic GMP. So color vision is affected, not they can still see. So that is actually a warning for pilots. I did not know that, but I just thought it was fascinating how a medicine. And by the way, that you may recognize that this medicine, sildenafil, was actually used, investigated, I think, by a small company, for cardiac use, because it is a vasodilator, so it may help relieve chest pain and may be useful in lowering a rare but very fatal condition called pulmonary hypertension. Means your blood pressure inside your artery that takes blood to your lungs may go up. But the patient, the phase 2 trial people complained to the doctor, says, by the way, doctor, we're getting erection. And Pfizer rightly decided there was a lot more money to be made in marketing as sildenafil for Viagra, and the floodgates open up. And there was a very humorous movie made about a salesman for Viagra. I forget the name of it, but was absolutely hilarious. That how that the medical establishment welcomed this salesman for samples. So I forget the name of the movie, but I think it was starring Ryan Gosling or somebody, but it's absolutely funny movie type of thing for those of you who want to uh, indulge in it. Okay, so that's Crystal Gale for you. And now this can be very serious also. It's, we can laugh at the Viagra, it's a temporary side effects, but one mutation can cause, okay, one migrant to a small island in Micronesia, I think he was Portuguese or something, had, lots of kids, and then a lot of almost in the Micronesia, this island's name is Pingala, but it's 5% of people have colorblind. I mean, that is just scary type of thing. And the great Oliver Sacks, I'm sure most of you know about Oliver Sacks, but he was the man behind the well-known Robin Williams movie Awakening, which was the discovery of L-Dopa at a Bellevue Hospital in New York City. Okay, and he is obviously a very erudite, popular neuroscientist. Okay, and so this is again, it can be, and all this became aware with the starting of train traffic, because obviously that was a big, well-known accident that took place in 1875 in Sweden. Okay, and finally the physicians decided to test the railway engine drivers. About 13% of people were colorblind, and so that became a really a big requirement for screening people, because when they couldn't see the lights. Okay, so this is, again, the ion channels as to how the smallest change at a small level becomes important. Okay, we still have 6 minutes before we take a break, I guess. Okay, now this can be, a boon or a burst, so this was an insensitivity to pain. Okay, you can say, well, that's a great thing. If you don't feel pain, no, we should also, try to have that mutation. Okay, but that is not so great afterwards. There was a cluster of family that was reported in Pakistan, because we may have talked about in the genetics class, in a lot of the Islamic cultures, to preserve land, people marry their first cousins. So even a somewhat recessive mutation, which wouldn't be that much of a problem, becomes a serious issue. Okay, and when you marry your first cousins, okay, type thing, and so there's a whole cluster of people that develop this insensitivity to pain. Okay, and unfortunately, due to poverty, some of the kids decided to make money by jumping from a wall and walking on fire, and unfortunately, eventually, some of them lost their lives, by using that as a stunt. Okay, and this again boils down to the sodium channel abnormalities. Okay, so, and this has been cloned down in families, and was located to a same part of chromosome which had the mutation. Okay, so, and I have extensive notes for the people who are a big genetics fan type of thing. But and it again, the point is that this channels interferes with the spinal cord ganglion or the nerve cells. Okay, that functions as nociceptor. Noci means nocic stimulant from the periphery that comes around. Okay, and this is one of the channel that has to do with the puffer fish as well. So how all this web is interconnected is interesting. So this is insensitivity to pain that has been can be a problem. Okay, and again, this the science has gradually advances, and the touch and pain channel, this was recognized as recently as 2021 by David Julius and an Armenian scientist, Arden Pataputian. Okay, I'm mispronouncing it obviously, but this is the touch and temperature, heat, and okay, you can see. And he used a lowly no, a red chili, and worked on the DNA fragments as the receptors and discovered these channels, the touch and pain channel. Okay. Okay, and let's see, similarly, this is the pressure things that I talked to you about earlier, how, and again, they were able to not just vent, but they were able to clone the genes. Okay. And so varieties of receptors for red chili and pressure sensation. So that was duly recognized by the gnomes in Sweden. Okay. This should, oh no, this didn't play nicely. It should have. Okay, now let me go back. Sorry, this is not doing as what it was supposed to do. Okay, the picture behind this, I'm sorry, but I, did not do my, animation correctly. Okay, this is a picture of the human ovum and all these little things that we see. I'll share the picture again. These are all the sperms that are trying to enter. Okay, so this is, you can say that this is your selfie. This is what you and I all looked like in the beginning, okay, but only one sperm can enter, and that again has to do with varieties of channels. And this is, now the ovum is not exactly, the female egg is not exactly a passive receptor as to the one sperm that swims faster and has higher motility can get in, but it selects the actual sperm based on the health, based on varieties of hormones, progesterone, and again, that, and so this is, and this again, in the testes, there is an ion channel that has been named cat sperm because it may have been recepted from in the spermatozoa of cats or something. So that's how the name came about. Okay, and was discovered in 2001. It has to do with sperm motility, and so it is a complex of varieties of proteins, as you can see how. Okay, sorry. Okay, so the calcium, ATP is the good old thing, and cat spur channel. You can see the picture as to how intricately evolution has used all the things. So, and so this is, it discovered in, so, and by after this, now interestingly, one of the species of Drosophila, the fruit fly, has a giant sperm actually that crawls in, it is huge, like it plugs a female trap. So that does not need that channel. So it's interesting how nature just uses the use. So cat spur channel is called calcium specific channel inside the sperm. So you can have a fun talking at the party about this as to how how sperm and ovens are selected. Okay, so it's not just the looks, but even down at the cellular egg level, women have choices. Okay, I think we're coming up to 11 o'clock, so we should stop for a few minutes. I think I will stop this share if you wouldn't mind pausing the recording.
Can you hear me? I can't hear you.
Yeah, I'm sorry, I was muted. I was playing around with it. Yeah, go ahead. Now I can hear you.
Oh, that's okay. I thought you were on and I didn't hear you.
Oh yeah,
That's
no,
okay.
I'm just trying to optimize my.
All very fascinating. That's all. It's really cool.
Okay, good. Thank you for saying that. Yeah, appreciate it. Okay, yeah, I think are you able to see the slide now?
Yes.
Okay, good. Yeah. Okay, yeah. So, all right, good. So I think now I'm finally got it figured out. Because and you're not seeing my notes also, right? No.
No.
Okay, good. Yeah. So we're in business. I think this time I was doing something weird that time. So, okay. Because you have to set up the presenter view before you start sharing, so that was my problem, so I think I hadn't set it up. Okay, I think, we should probably start it far less stressful than the biochemistry because, but if 1,000 students join Wash U and St. Louis for pre-med, only 100 get into, well, finally, only half of them stick around because they all drop out because they don't like biochemistry. So that's no wonder it's kind of okay. I don't know if our biochemist John is here or not. I don't have the list, but I think, it's a challenging field, and I can recall to my days of biochemistry that I always used to skip that lecture and, because it was so dry and unfortunate. It was always at 11:30 when we would rather go to the cafeteria rather than listen to all these amino acids, But it takes a special kind of person to be a biochemist, but thank goodness that they do. Because, and okay, now this is not just that. So we talked about at conception how the ion channels played the role. Okay, now moving on to further after maturation, ion channels at birth. Okay, we need them right when as soon as we get out of our mom's womb. Okay, and take a breath because the epithelial cells of your lungs, have channels, and these channels have the capability of drying out all the mucus with air. Okay, and that's why if you are born a little too early, that does not turn out to be good because you get the premature lung. Okay, and so this is why, and there's a lot of the deaths that occurred because the lungs were filling up with the newborns, and finally, back in the late 50s and early 60s, a chemical called surfactant was discovered, and that saved a lot of premature babies' life. So again, so this is, the aquaporin. Remember we talked about the channel. So the real science comes into the practice, and inside the lung cells, the water absorption and the sodium and the chloride channel, they all play the role. Okay, it's a complex mechanism that takes place. Okay, and so this is, and this is again mom's stress hormones at the time of delivery and labor. They play a role, and a lot of these ion channels have a role in kidneys and hypertension. Okay, and if there are defects of the ion channels, that can also cause defect in something called the neural crest defect. Okay, neural crest is the primitive area where your spinal cord and eventually brain develops. The nerve cells migrate. The early embryonic development. Okay, now it turns out that if you have fever, okay, that can harm this neural crest cells. Okay, in the first trimester. Okay, and this channels can interfere with the development of neural crest. Okay, and so that's going to be interesting to see with the recent recommendation of not taking Tylenol as to how the basic biosense, that having fever during early pregnancy may not turn out. We may increase more birth defect of cells, as to by interfering with the neural crest cells and the ion channel. So I just had a paper which I did not put the reference because it was rather technical as to how the fever can affect the neurocrest ion channels. Okay, the interconnected is fascinating. Okay, and again, this is the example of the cystic fibrosis of the premature babies. Okay, the normal airways. Okay, the air mucus, it's migrated and it's interstitial fluid inside the lung. And in cystic fibrosis, all the mucus sticks around because of the abnormal mutation of the ion channels. And this has been a classic detective story of the cystic fibrosis has been discovered. And now finally there are some highly complex specific drugs that target the mutation of this cystic fibrosis ion channels. And that has made a dramatic difference. But it only works for the, unfortunately, for the rare mutation that takes place, not for the common kind. So again, we haven't totally, so classic example of ion channel mutation. And this goes back to the early days before science advanced, that the salty forehead of a child, so the midwives would lick the salt or the sweat of a child, that if you have a salty forehead, the child is likely to die soon. It's cursed or something. So, okay. And this discovery was done, during the hot summer in New York City in 1950s by a physician who's with a complicated name of Paul D. San Agassi. Okay, there are a lot of people who are dying, and the cystic fibrosis people were dying a lot more in that test. And that's when he discovered the test for estimating the sweat chloride. And so the test to diagnose cystic fibrosis came into vogue, in the 50s. Okay, before a lot of this biochemistry and genetic mechanism was discovered or elucidated. Okay. Okay, so every breath you take, so remember the rock music fans may remember this song. Okay, so it's, how the basic science ties into healthcare and health medicine. And an interesting book written by Dame Frances Ashcroft. Okay, now that it's, people may recognize OBE, Order of the British Empire. Okay, the empire may be gone, but they still hand out the revolts. And now this is, I had to look up what DBE stands for. A Dame of British Empire. Okay, and this is Fellow of the Medical Sciences, and the FRS is the Fellow of the Royal Society. So these are the abbreviation of Dame Frances Ashcroft. And she was a very reputed researcher of ion channels, and based on, and she was interested in diabetes of the newborn, and discovered a mutation. Usually, diabetes occurs at a later age when you start taking more glucose and sugar. Okay, so it's diagnosed at a later age, but the diabetes of newborn can be a serious thing. And it turns out that has not to do with, the deficiency of insulin, but it is related to mutation in one of the ion channels. And based on this, she was able to save a lot of painful treatment with insulin. That this potassium channel abnormalities can be treated with simple medicine called sulfonylurea, which is your metformin, the cheap drug. Okay, so usually the juvenile diabetes requires insulin. Okay, but this was an exception, that the special kind of diabetes of newborn, which is due to this mutation of ion channel, can be treated with drugs. So it saved a lot of misery and stress to the babies and to their parents, obviously. Okay, as to how fascinating is how and this is, okay, let's see, I hope this plays now. This is the goats video that is.
There are dozens of fainting goat videos on the web, but we wanted to find the nerve center of the fainting goat world, which, as it turns out, is in Florida. Meet Terry Linacci of the International Fainting Goat Association. She knows exactly how to make a goat faint.
What usually triggers the fainting is when they're startled or even excited, they will faint. They'll fall over or stiffen up when we feed them in the morning. They get excited. And also, we've seen the bucks faint during mating seasons. I think it's the adrenaline rush of sex gets them, I guess. Typical guy. There's a good one. Go! And it's confusing because they don't actually faint. They're fully conscious. It doesn't hurt them at all. They stay down for maybe 10 or 15 seconds, and they can hear, they can see.
Ah, great fun for humans. But what exactly makes these goats do a full-on face plant?
Fainting goats have the condition myotonia congenita. It's a genetic condition affecting a particular gene telling muscles to tense and to reset, to relax again afterwards. Normally, they relax instantly and we're able to flee or fight, whatever we choose to do to defend ourselves. In this case, the signals from the brain are sending through the message to tense all the muscles. But because of this genetic condition, the signals telling the muscles to relax again aren't getting through. And so it tenses up, can't run away, and falls over.
Some farmers used to use these myotonic goats as decoys. If a wolf or some other predator were to attack the herd, of course, the myotonic goat is going to go down, and that's what's going to take the predator's attention away from the rest of the herd. That's not very nice.
And because all mammals' muscles have similar chemistry, many species, including us, are prone to myotonia congenita. Are your thoughts enough to stop you in your tracks?
Okay, so and so varieties of diseases, some of them, are cardiac and which are more close to my heart. Ouch. So that I'll talk more about it as we go along. Okay, and this, some of these are very serious and fatal, and I have memories from my personal experience on that. Okay, and I already showed you this slide, so sorry about the repeat, but this was like I should have eliminated that. So, okay. And then again, the prototype of the things of the ion channel listing, brainstorming. Okay, brainstorming is nice, but when the brain decides to hold a brainstorming session on its own, that doesn't turn out to be so good. And this has long historical thing, starting from the eras of Hippocrates. This was considered as a sacred disease, and I think as a starting medical student, I saw Cleopatra, which was the classic demonstration of Rex Harrison's epileptic fit that Elizabeth Taylor puts in the tongue biter in his mouth. So that was the first classic demonstration. I think I was probably 12, 15 or 16 when I saw that. Okay, so this was the mechanism. So epilepsy has been, maligned as, a bad omen and witchcraft and the grand tonic seizure. It is a historical root. Okay, and so the exciter, so the brain just goes berserk literally, and it causes lots of changes in the ion channels and results in specific area which causes, a focus, and so varieties of epilepsy, and so you could just almost specialize in epilepsy. And or one of our mutual Ollie friends, Peter's wife, who passed away, she was a specialist in pediatric epilepsy, and so it's a serious a problem. And there are varieties of seizures, tonic and clonic seizures, and the so-called grand mal seizures. And varieties of myoclonics means only a certain group of muscles, few muscles, okay. And there can be absence seizures, which is just a slight eyes blinking. So you may feel like you're awake, but you are having a seizure sitting around, So the loss of muscle, so it comes in all sorts of forms, type of thing. So, and it has led to varieties of advances in medicine. Okay.
Brett, it looks like the slides have stopped.
I'm sorry. Oh,
Can you tell me, because my slide says ion channels at birth.
Oh, okay, let's see, share.
Yeah, mine too. Yeah.
Oh, I'm sorry.
Mine 3. I couldn't see the movie. I could hear the movie, but it's stuck on ion channels and it shows your little box if you want to pick a presentation, a screen, a help, an end show. And that's all I've got.
Okay, yeah, thanks for pointing out. Yeah, I'm sorry, I should have.
I thought it was by computer.
I
Well,
signed it. I signed in and out 3 times, but it didn't help.
Oh,
I
yeah.
signed in and out of the meeting.
Let's go, yeah. there we
Oh,
go.
God.
Go back.
Anyway,
Okay, ion channel set. So which one are you seeing at frozen at?
Nothing right now. You're not in presenter mode.
Oh, okay, yeah, just a second, okay. Share. Okay, you should have this now.
Ion channel drugs.
Okay. Okay, got it now?
Yes, that's what we have, but you just put your little box back in there where you're picking stuff. I think that's what freezes the screen.
Yeah, I think your cursor's in the middle of your screen.
Okay, all right, so stop share and let's see. Okay, you're back to the ion channel, right? Yeah.
Yes.
and channel jokes. Oh, popped up again. There it goes.
Okay, so we talked about epilepsy. Okay, now you can see the slide, right, for epilepsy?
Yes.
Yes.
Okay. All right, so this is when the brain decides to, and I showed about the varieties of types of epilepsy, the tonic, clonic, and myoclonic. Sometimes you can just see where you feel like your eyes are open and you're still having seizure and epilepsy. So now the treatment, okay, came into existence like as early as 1908. Okay, now the German chemical industry was in the vogue after they took over from the Brits. As you may remember, we talked about Michael Faraday, the great Michael Faraday, who was a pioneer of the alternating current. Okay, did also the chemical experiments. Okay, where he was the first one to discover the chemical structure of benzene. Okay, you remember the benzene molecule, which is the workhorse of coal tar industry, and that led to the rise of German chemical industry, and they went on to develop a purple dye and put it started the big revolution of chemical industry and coal tar industry, and that led to. But again, so that was the start of Michael Faraday and the rise. So on the tails of that, some of the first chemicals that were synthesized were, in the early 20th century, were barbiturates, is used for sedation. Okay, and then again, the starts was resynthesized by a well-known company that is no longer in existence called Park Davis. Okay, and this, now they've all been incorporated and perhaps was absorbed by Pfizer, I think, okay. And so this was, again, the dilantin later on was discovered, and it worked. It was a chemical. It was just based on trial and error. But then later on, we learned about the chemical mechanism of voltage-gated ion channels, and that's where it works. And in the early days of cardiology, we also used for treat abnormal heart rhythm. And obviously now a lot more powerful drugs have become available, so that is no longer used. So it is explored in varieties of diseases. Some are legit, some are so-called off-label use used by medical community. Okay. Okay, and this brings back to the memory type of thing. When I was in practice in 1980s, I got a book, without subscribing for it. This was a big book, type of thing that was sent to all the practicing physicians in United States. I don't know if Dr. Bruchette is signed up or not, but Dr. Bruchette is a friend, and he usually he usually watches this afterwards, so I don't think he's here, but he may have received this book also, that we all received this book. Obviously there are, thousands and thousands of physicians in the United States, but this was a book that was sent by Jack Dreyfus, and nothing to do with the French, a Jewish, a guy who was the cause celebre during the late 19th century, who was accused, the Dreyfus affair, that Bruce Gobioff did a class. But this Dreyfus is the Dreyfus of the Dreyfus Financial Fund. Okay, he was a multimillionaire, and he spent his own $70 million of his own money to send out this book. This was a wonderful drug that improved anger and depression, and he even supplied it to good old Richard Nixon. Okay, and so, but that again, so he was promoting this drug as a cure all for a lot of mental illnesses. So it goes to show you. Okay, so this was, but obviously, that didn't turn out to be. He spent a lot of money, so I still had that book for a long time on my shelf just for curiosity. Okay, what nonsense is this? When I read this, I said, this doesn't make sense, it had because by that time we had a lot more other sophisticated medicine for cardiac rhythm disturbances, and as well as for neurological illnesses. Okay, and the side effects of Dilantin are nothing to sneeze at. Your gums swell up, and you get a lot of other side effects as well. So it's not as benign as one might treat to be. Now, this is sometime, my wife is a retired anesthesiologist, and she recalls that this was a big nightmare for them. They would all be dreading that sometimes, as a effect of anesthetic agent, which is a common gas, if people have abnormalities of their channels, okay, for the calcium, it causes, abnormal calcium release and causes abnormality of thermogenin protein called thermogenesis. And these are the receptors, this is RYR is a ryanidine receptor, which just ignore the name, that has to do with a plant that was used as a model for this channel. And so it causes massive temperature rise. All of a sudden, soon after anesthesia, the temperature can go up to 107, 108 in Fahrenheit. And that is a rare illness, but 1 in 20,000, but it can be fatal. Okay. And it is also, by the way, we are related to pigs. So even in pigs sometimes can cause with stress. So it has to be properly treated with a very uncommon drug, which not all hospitals have in stock. And so that can be a real nightmare. And turns out as to how selective breeding can be a problem, that there is a small caste of Brahmins in one part of India, which have there is a lot of intermarriage that occurs, and they have very high incidence of this malignant hyperthermia. So it is interesting that when you're admitted, a weird question would come, what caste are you? Okay, but there is a scientific reason for it, Okay, so the malignant hyperthermia, it's really a nightmare illness that is related, again, the biochemical cause that plays a role. Okay, now we talked about mutations, obviously, these are all interesting. There's things that this mutation that takes place even in horses, that and it is linked to the quarter horses, and it has to do with potassium, and the mutation causes muscle spasm and trembling in the horses. Okay, these are the well-developed muscles, so that is one way to game the system by breeding horses. Okay, And so that's another mutation that takes place, inside the horses, again related to channel abnormalities. Okay. And again, you can just see this is the scientific basis that takes place of the muscles, I think. But we have talked about, how the thick and thin muscle fibers that takes place at the entry of the calcium is introduced, and the muscle fibers tighten up. The same thing that they talked about, in goats, applies, to the bigger animals as well. Okay. And again, okay. I think I'm going to skip this one. This was not, okay, let me stop this share, and I think I'm still having. I'm again going to see if I can do the portion of a screen, Okay. Okay. Are you seeing a slide?
No,
Okay, yeah, let me just go to that.
No,
Okay, yeah, now you should have that. Okay,
Yeah, I see it now.
yes.
yeah, okay, I'm back in business, yeah. Yeah, let me just make that red square. Okay. Okay, let's skip this one. It's not. Okay, let me move my bar. Okay, now the varieties of uses of electricity have been used, or now we already talked about the brainwave test, and I've introduced the subject of, the dog and the rise of electrocardiogram that took place in 1924. We'll talk in more details about heart in next few batch. Okay, but there is also the ophthalmologists are not using as well. Okay, you can say, well, they have actual direct window, they can see the eye, but still it is useful to record electricity from the eye. See the different, the electrooculogram. And so that's discovers the function of the retina and the retinal epithelium. Okay, and it's useful for diagnosing. You can just see how the films are recorded, with the eyes moving, and they can record the electrical activity of the eye. So that is useful for them about differences between the cornea and the varieties of tiny membranes, and the voltage difference is exquisitely small, 6 millivolts, So this came out in the 1950s, okay? And so this is varieties of information about the ratio and the health or disease of the eye epithelium. So eye doctors. Now magnetic cardiogram, because along with this, you can improve the sensitivity of the brain waves by using magnetic electrogram, mapping the magnetic field of the brain currents. Because any current, generate not only the electrical field, but also magnetic field. Now this is very high tech, okay, because it requires powerful magnet and electric current, and the sensitivity of magnetic field is relatively small, so it's used only for, it has never, and the bigger use of that has evolved is in a technique that you may even have had the test called MRI. okay, which has to do with the water molecules and changing the radio field, the position of the hydrogen ions inside the water molecule in your body, and recording the position of the molecules after and very complicated mathematics that is involved in MRI scanning. Okay, all right. And now this, I'm going to wait for this one because I'm going to talk about this as to people who have done experiments on themselves with brain-computer interfaces. And this was a, a neurologist by them, a neuroscientist called Kennedy in Georgia, and he wanted to do research on the brain-computer interfaces. But this was in the early 80s, and nobody wanted to do this. So he decided to volunteer himself and put under his knife, and went either to one of the South American countries and got a brain-computer interfaces called neurotropic electrode implanted in his own brain, attached to the computer. The surgery was rather complicated, and this was another way of interfacing electricity with human brain. So some of the earlier attempts that were done, and he nearly died from it, and I remember reading this story in one of the computer magazines, I was saying, so it just goes back to the early days of John Hunter, or the guy who did the heart catheterization. John Hunter, by the way, during the early 17th century, infected himself with syphilis. And you are familiar with heart catheterization, where the German scientist inserted the heart catheter in his own body and took the X-ray picture in the 1930s. And that was the start of coronary angiography. And as to start with, okay, so self-experimentation has been around. And the most important self-experimentation that you may or may not know was the H. pylori, where the bacteria can cause stomach ulcer. And there was the Australian scientist who drank the cocktail of the bacteria themselves, and sure enough got the ulcer. Okay, so that change was the so-called paradigm shift in medicine. Okay, all right. And this is, now the brain science has been used. This was, the brain-computer interfaces were dramatically demonstrated by a physician in 1965, okay, named Dr. Delgado. Okay, obviously, he was Spanish, and experimented the bullfighting ring in Cordova. Okay, he was a professor of medicine at Yale University, and again implanted the radio transmitter receiver inside a bull's brain. Okay, and the chip was altering the electrical signal with radio signal, and again, and stepped into the ring himself, and the charging bull, by pressing the radio signal, stopped in its track. Okay, so this was a dramatic demonstration of electricity and brain. Was done by late Dr. Delgado. Okay, and he was a very reputed scientist, okay, and he was able to remotely control the emotional behavior of various animals. Okay, and so that segues into the controlling the behavior of humans. And obviously, right on his coattails, the U.S. military was involved in this thing, using for military purposes and fighting the bad commies. Okay. Okay, and I think we'll skip this one. This is a little too technical. So this is the varieties of medicines that are, okay. Okay, and now after we talked about the eyes, okay, the same way, or another importance is hearing also involves the use of electricity, the sound waves that come into our ear, the external ear, and in the eardrum and transferred inside the cochlea. And there are varieties of exquisite, you can see this electron microscope, the tiny hair cells, okay, that are directional oriented and they move along with the sound waves, okay, and they generate the electrical nerve potential and signal that goes to your auditory area of your brain, or which occurs on the side over your temples, and it's converted to electrical signals. Okay, and so these are the varieties of. Okay, up. Let's go back one more time. Okay, and see if I can show this. Yeah, you guys can see how the once I magnify it, so the membrane inside this that vibrates like a drum and causes the movement of the hair cells. Okay. And so this was this is how the signal is transmitted. Okay, and hope this one plays.
If we zoom right the way in with an electron microscope, we should see something remarkable. The secret of how sound is transmitted to the brain. They're rows of minute hairs, only a few thousandths of a millimeter high. As noise vibrates them, they send electrical signals to the brain, which we experience as sound. It's fascinating to watch, and we're about to see it happen. Take a closer look at these V-shapes. They are, in fact, clusters of 3 lines of hairs, which are part of a built-in amplifying system. Now, if we take a look below, we should see the rest of the amplifier. There we are. The hairs are sticking out of a cell underneath, shaped like a sausage. It's actually been possible to isolate one of these hair cells, so let's see what it does when we play it some music. You might recognize the tune.
1, 2, 3 o'clock, 4 o'clock, rock. 5, 6, 7 o'clock, 8 o'clock, rock. 9, 10, 11 o'clock, 12 o'clock, rock. We're gonna rock around the clock tonight.
The joints jumping, literally. The excited response of these hair cells amplify the faint vibrations that arrive here from the outside world. And they do it so well that we can actually hear the sound of a pin drop. Unfortunately, from the moment we are born,
Okay, we only have a limited number of hair cells, so that just goes, it's a long video. Okay, so once you lose it, you lost it. So if you are listening to, hard rock at a younger age, you're going to end up with a lot of hearing. So that's a huge thing. And I think one of the professors from School of Public Health did a class at Lake Magdalene. So if he does a class again, it was amazing class on audiology. So they brought in a lot of the equipment and are recruiting people for the various trials for the equipment as well. Okay, so it was an interesting class. I don't remember the professor's name, but that was really one of the nice classes. That he and his audiologist, they both came and did the class a few summers ago at Lake Magdalene. So keep an eye on it if they're doing it again. Okay, and this is okay.
A cochlear implant is an electronic device that can restore sound perception in people with certain types of severe hearing loss by picking up sound from the environment, converting that sound into electrical impulses, and transmitting those impulses directly to the auditory nerve. Cochlear implants provide an option for hearing restoration in patients who have more extensive hearing loss and for whom a device that simply amplifies sound, such as a hearing aid, may not be as effective. A cochlear implant is an example of a neural prosthesis, as it replaces the disrupted functionality of the cochlea with an electronic device to restore functional hearing. Cochlear implants consist of both external and internal components. The external component typically sits behind the ear and picks up sound through a microphone. When sound is detected, a sound processor converts the auditory information into a radio frequency signal. The signal is transmitted to a receiver implanted under the skin behind the ear. The receiver decodes the signal, then converts it into electrical currents, which are sent along wires that have been surgically inserted into the cochlea. The wires stimulate the auditory nerve, a job typically reserved for the damaged cochlear cells, and stimulation of the nerve causes auditory information to be sent to the brain to create sound perception. A cochlear implant does require surgery, but major complications are uncommon. Studies have generally found cochlear implants to be beneficial, but the extent of the benefit does vary depending on the patient, with children who get cochlear implants earlier and adults who get them soon after hearing loss typically experiencing the greatest benefits.
Okay, so we saw that. Now again, remember that we are using electrons, okay? We are using current that gets in our house. We are not using biological current. And when we are putting in wires, you're still likely to get tissue reaction and other things. So we haven't solved the basic testing. We are not getting down to the scale. Interestingly, just yesterday, I saw a report that with the so-called computer-generated neurons, Amherst College, they were able to load down to get a response at 0.1 millivolts to and something called brain organoid. So in other words, they were able to connect a transistor type device with a tiny voltage to work with biological system. So we're able to scale down dramatically. So that really may have a bigger advantage if it becomes more feasible or scaled down, not scaled up, obviously. Okay, so this is, again, and that has been the problem of the electrodes causing problem with the tissue reaction. Brain does not take too well to any type of metal being inserted inside your okay.
Deep brain stimulation is a minimally invasive procedure. It can be effective at treating conditions such as essential tremor, Parkinson's disease, and epilepsy. The idea is that we use the brain's natural language, which is electricity. And instead of making the brain talk in a particular way, we're disrupting the brain from talking in an abnormal way. The first part of the procedure is where we implant the actual electrodes into a very specific nucleus in the brain. Typically, patients have it done on both sides to treat symptoms on both sides of the body, but it doesn't have to be that way. It can also be one-sided. If it's both sides of the brain that we're treating, then we would make a small incision on both sides of the scalp. It's usually about 2.5 centimeters long, and make an opening in the bone about the size of a dime. And we use a very precise apparatus to gently guide the electrode through the brain tissue and down into that target nucleus. We use microelectrode recording to actually listen to the brain cells. And by listening to the brain cells on the way down, I can actually tell exactly where we are in the brain. And then we can finish up with that first procedure by just taking the electrodes that are now in the brain, and the back end of those electrodes, we just pocket them behind the ear, underneath the scalp. And the total procedure takes somewhere around 2.5 to 3 hours. Then we bring them back for the second stage, which can be anywhere from a few days to maybe a few weeks later. And the second stage is where we take the back end of those wires, which we tucked underneath the scalp. We just make a small opening right on top of those wires, and then make an incision in the chest where the stimulator itself will go, which is kind of like a pacemaker, but for the brain. And then we create a pathway underneath the skin to connect from the top of the head to the chest. And that procedure itself takes about 30 minutes and is an outpatient procedure. There's two things that I hear from patients post-operatively. It's new lease on life, and I wish I had done it sooner.
Okay, so that's a small school on the West Coast, but some of the videos are really dramatic, but I just wanted to show the procedure as to how people can really, from shaking like a leaf during a storm, people would stop absolutely and have a normal conversation. So it's really a groundbreaking advance in electrophysiology. Okay, and similarly, and this was again, at the Gosh Hospital, which stands for Great Ormond Street Hospital, a well-known institution in UK, have had an implanted device for epileptic seizures, And again, this was in the thalamic area. These were again inserted the same way which was done. Okay, so the electricity and brain are really being used, widely used, Last but not the least, letharism, which occurs, another type of poisoning that takes from a plant. This was mentioned as early as 400 BC in the Ayurveda tradition, and was mentioned by Hippocrates in Greece, as well as, and this was rediscovered, so to speak, and put on more scientific basis in the 19th century by British officers working in India, where they saw this type of paralysis that occurred in cattle as well as in human ingestion of this type of Lethrus sativa. Okay, that again is a neurotoxin that took place, occurs in extremities and causes paraplegia. Okay, and can cause death and deformities. Okay, so again, plants can be tricky. Okay, and this shows the more animal letherism, and this is the particular poison that takes place. During personality. So varieties of symptoms that can occur. Okay, and in human letherism, you can just see how deformed this poor chap is. Okay, and this is okay, and there are advantages, and people are working with trying to get rid of the toxic effect out. Okay, now I thought this was an inherent story. I mean, all of us are old enough to remember the great movie The Birds. Okay, which was, I still can't figure out how it did that, but there was some marvel of movie making, and there may have been some science, that birds falling from the sky, they were vomiting fish. Okay, and this occurred in a small town in California. The shearwater birds, they were eating fish that was high on a phytoplankton, which had a chemical called domoic acid, and that messed with the glutamic receptors and worked with the calcium, this thing. So that's one of the reasons why the birds were acting weird. So there may have been some scientific basis behind the, obviously he did not film this during uh, the season that was occurring, and but so there may have been some, they may have read the story and that led to the movies, so as to how science at the movies was. Okay, and again, we already talked about the stiff baby syndrome. Okay, that takes place again, varieties of channels. So I think I'm beleaguering the point, so I think, and this again, plays a role in the Agatha Christie strychnine poisoning, the very first novel that she wrote, and A Mysterious Affair at Styles. So that's where it figures loudly into it. Interestingly, a fruit that is in the Southeast Asia that is used miraculously, okay, that is varieties of fruit that messes with your channels on your taste buds, okay, via ion channels, okay. So that can again, can cause toxic effects, So this is, and okay, so it's a lot more detail than you prep. And this is important. This is a problem in the Netherlands, what's more, and it can occur even here, as to people who overdose on black licorice, and that can mess up with your ion channels again and cause low potassium with a chemical that becomes part of the licorice called glycolic acid, and that can cause almost a cardiac arrest, And this was a case report in the New England Journal in 2020. And again, I was listening to talk about ion channels, and somehow in Netherlands, this is a big problem, type thing. So there was even talk about whether they should put a warning on the black licorice. So be careful as to how, it can see this is a compound that works around, again, it's it goes inside your kidneys, and kidneys have a lot to do with your preserving your potassium and sodium, as it is filtered and reabsorbed, so which is inside the tubules, and it goes back into the circulation. So that is where this metabolite of licorice can cause a problem. Okay, let's see how we still have some time. Okay, so that can be. Now I think we'll start out with the heart. Okay, I think, oh, wait a minute. I think somehow this became bigger. Are you seeing the small box or the big box?
It's a big box. It says the beat goes on.
Yeah. Okay.
But I got,
Yeah, I'll go back to it. Yeah, I think, this is what, yeah, I think somehow it got defaulted to. Okay, now we should be in business. Okay, I think the green doesn't like the green. So now you should just see the slide. Okay.
Yep.
Yeah,
Yep.
all right.
Got
Now
it.
now we're in business. So the meet goes on. So we talk about the Waller and Jimmy the dog. Okay. And this is about electricity and heart are even more connected than the brain and electricity, okay, because, if you don't have a beat, that's the end of it, so to speak. And these are that different in the pacemaker cells of the heart, that takes place, like unlike the other action potential in the nerves that you remember from that, well, after the action potential dies down, it returns to the baseline. Heart has something called automaticity, okay? So it has, sort of calcium channel. So you can see how the line is different. It's not flat. It rises by itself, and that has to do with it is a special channel that has a principle of automaticity, means the impulse starts on its own, basically, okay? And so that's what creates life and heartbeat, okay? So there are slower depolarization and lower peak, okay, that takes place, and this is the so-called pacemaker cells inside the heart. So that is what keeps you and I alive. On the other hand, the muscle, the cardiac myocytes, okay, it's a little different. That has more sodium channels, okay, and different calcium channels. So impulse just goes up, goes down very quickly, fast, higher peak, So the difference between so-called pacemaker cells of the heart and the cardiac myocytes. So this is what keeps you alive, so to speak. So there is a totally different, and this was called the so-called funny channel that was discovered, as it gives the real underlying biological or molecular mechanism of your heartbeat. Okay, so this is what really keeps us alive. Okay, and we already showed you this. The Einstein discovered the electrocardiogram. He amplified on the work that was done initially by the Brits, which was a very weak signal. And with the German patience and thoroughness, he developed a galvanometer, which was 600 pounds. And this poor gentleman is putting his feet inside salt water, and the electrical signal is recorded and amplified, and on something called the string galvanometer. Remember Galvani, which we talked about, and he discovered the patterns of the heartbeat. Okay, and the various patterns were identified, and he labeled them in proper sequence. And this is, the action potential of the pacemakers. As you can see, the spontaneous depolarization. This is what the real secret of heartbeat is. Okay. You can see action phase 0, depolarization. Okay, calcium channels are open, rapid influx of calcium that takes place. Okay, triggers action potential. Again, phase 3, calcium channels close, potassium channels open, and efflux of the cells. And you can see how the line is the fourth. Okay. And these are, unlike other muscle cells, the heart cells are connected with something called the gap junction. So they're all in continuous. The technical word for that is called syncytium, continuation. So there are varieties of discs that allow the heart muscle to contract in a wave type of pattern. So the heart can work as a pump. So there's a nice, these little things that are here in the middle are the intercalated discs, and it is tried to organize and nice bundles, basically similarly, but they're interconnected with each other, and that's how they can beat in synchrony. Okay, all right. So so the whole muscle works in unison. Basically, that's the thing that takes place. So the impulse, the action potential that takes place, it's transmitted down and in a nice coordinated fashion inside the entire heart muscles. Okay. and this is, a more detailed diagram of there are varieties of timed clocks that takes place inside the heart, coupled clocks of inside the pacemakers. You can see there is the calcium influx that takes place, and this is called depolarization and repolarization that takes place, which I showed you earlier, basically. And because this calcium channel was so unknown, the people who are researching that labeled that as a so-called funny channel. And this is just totally ignored. That this is almost, I got a little carried away. It's a lot more complicated biochemistry of the varieties of currents that takes place. And only if you are a true biochemist at heart, you would be interested in the inward sodium current and potassium current and the varieties of proteins that have a molecular dance that we can take that takes place. Okay, so I think we are so just ignore this unless you are really into some heavy biological sciences. And well, okay, let's see how we are doing with time wise. I think I have about I should be able to finish, so we can stop here about the problem, which is more interesting, about the sudden death in athletes and art, and we'll continue with this at next time, because the basic physiology of that is, again, involves all the ion channels that we talked about. So they are useful at all levels, from individual cells to the most important organ, i.e. brain and heart. And we have taken advantage of these properties in curing the illnesses that affect us, not just with Parkinson's or cochlear ear, but it has a dramatic role in cardiology that we'll address next time. And hopefully we'll talk something about the regenerative medicine and advances that are taking place. Okay, and the so-called brain-computer interfaces that is being. Okay, so I have about let's see, about 60 or 70 slides, but some of them are videos which I may just send you the link. So we should be able to wrap it up next time. Okay, thank you, folks. It was a pleasure and I enjoyed it. Hopefully some of you did too. Okay, cheers.
Okay, thank you.
Thank you.
Hopefully it was fun.